We've now experienced the awesome animation through the Virtual Cell Animation Collection. And my goal for this short podcast is to zoom in and look a little bit closer at this process that Crick called the central dogma of gene transfer. That is the process by which DNA is converted to messenger RNA, and then messenger RNA can be used to make protein product. So in our virtual cell video, I want to draw your attention to a little ways in when we had zoomed in on the nucleus and we were looking at the DNA, and we were talking a bit about the process of transcription. So this purple molecule here is DNA. And so we're going to zoom in on that DNA and start by looking at that. So we've zoomed right in to that purple DNA strand, and oftentimes people call the DNA a single molecule, but in fact, as we zoom in here, you'll start to see an overlay. I drew this not using anything fancy like ChemDraw, but just a simple drawing program. I highly recommend it. I drew every single atom and every single bond, and it enabled me to start to see some of the inadequacies of trying to draw DNA in 2 dimensions. What we see in this zoom in, even with this distracting purple background, is that DNA, in fact, while people tend to call it a single molecule, is actually 2 molecules. There are 2 strands of DNA, and I'll draw your attention to those 2 strands. There's one here, and there's another one right here. Okay. So this is considered a double-stranded molecule. In fact, sometimes we speak about DNA as being dsDNA, or double-stranded DNA. And double-stranded is the way in which we tend to see DNA, although there are some viruses that have single-stranded DNA. So double-stranded 2 molecules, notice that the sugar phosphate backbone is held together by all covalent bonds. In fact, they're phosphodiester bonds. And then the 2 molecules are held by non-covalent forces, hydrogen bonds. We can see those in the dotted lines. Now let's get rid of this distracting background and look a little bit closer. I'm just going to shift the molecule, put it onto flat onto a page, which we will definitely recognize has a lot of inadequacies to try to look at DNA in that way. But what it does allow us to do is recognize the anti-parallel nature of DNA. So notice that what we've shown as the quote-unquote top strand right here, this is the 5' to 3' strand. Sometimes we call that the sense or the plus strand of DNA. And then, of course, by default, the negative strand or the 3' to 5' strand there on the bottom. We often actually get pretty lazy when thinking and talking about DNA, and biochemists tend to use a lot of shorthand. So oftentimes, rather than showing the full molecular structure, and don't worry, I'm going to bring this back, we will just show these 2 strands, the 5' to 3' and 3' to 5', in this very simplistic depiction like that. Of course, there's an awful lot, as you can see, that is implied when we do that, and we'll just go ahead and bring everything back, and we start to recognize all of this molecular structure. Now, the reason I said that I encourage you to sit down and spend, as I did, hours getting infuriated with trying to draw DNA onto a flat page, it simply does not work. No matter how much you try to use your techniques that you've learned in other chemistry courses to show stereochemistry around the bonds, you simply will find that it just doesn't pan out. And the reason for that is because the geometry of the bond angles is not It's not possible without the double-stranded or without the helical nature of the double strand. So in order to really start to see the true chemistry of the DNA molecule, a model is required. So here's a model that shows us a better look, and I think I like looking at it from the top down, but however you look at it, you can start to see the actual geometry of the molecule. Of course, double-stranded molecule. You can see how DNA has grooves in it. One of those grooves is larger than the other, so there's a major groove right there that's a larger groove. And then a minor groove that's a smaller groove. And you can see that the nitrogenous bases, their hydrogen bonds, when you look down, maybe from the top, you can start to see how planar those are. So it's almost as if they form steps on a spiral staircase. But what you also start to see is, rather than having an inadequate way of drawing the tetrahedral binding geometry at the carbon number 1, that you start to see in the model that it's easier to depict those that stereochemistry as the carbon bonds. And if I can show you to the nitrogenous bases there. So this double-stranded helical nature of DNA, the helical nature is what allows proper bonding geometry and gives the molecule its, of course, quintessential nature. So now that we understand a little bit more about this nucleic acid as a whole, so note that DNA is a nucleic acid. RNA is a nucleic acid. That is the type of macromolecule that it is. So that's important to keep in mind. The DNA and RNA are both comprised of repeating subunits. And you may have started to notice that. You might have started to notice that, in fact, one monomer after another monomer repeat in order to give this structure. So I've just grabbed a hold of one of these monomers, and I'm going to copy it, and then I'm going to paste it, and I'm going to turn it, and I moved it now over to the side. And so now what we're seeing is, if I make certain to add the hydroxyl, and now we have the building block of DNA. So this building block is called a nucleotide. So the nucleotide terminology, and let's go ahead and write that down, is used to describe any one of these building blocks of DNA that has a phosphate group attached at the 5' carbon. Note that the numbering on the nitrogenous base takes precedence, and so the numbering on the sugar ring is denoted by primes, so 1', 2', and I missed a number 2 there, so let's just steal one from here. And note that that's the 2' carbon there. So a nucleotide is the ribose sugar, the 5' phosphate, and the nitrogenous base at the 1' carbon. Now, technically, you could also call, so this is a monophosphate. There's a single phosphate that is bound there. And so you could have 3. When DNA is actually built, there are the building units that are used are nucleoside triphosphates. So they contain 3 phosphoryl groups bound at the 5' carbon, and that gives a lot of energy when a phosphodiester bond breaks. It gives off a lot of energy, and that can power the synthesis of DNA. So this building block of a nucleotide can start off with 3. Once it's in DNA, we can see the building block is a monophosphate. And so another name for a nucleotide is a nucleoside monophosphate. So this could also be called a nucleoside monophosphate. And so, of course, we've got this old bond hanging out that isn't there anymore. And a new negative charge, right, that would be present on that oxygen. So a nucleoside monophosphate, well, what does that indicate? Well, it indicates that the term nucleoside describes just the ribose sugar and the nitrogenous base. So we could actually go ahead and get rid of this phosphate, and this would just be a CH2OH. And so this is a nucleoside. So there are different naming strategies used for nucleotides and nucleosides. So if we're talking about this particular molecule, which the nitrogenous base that we see here is a G or a guanine, then this is the naming of the nucleoside that we've currently got drawn here would be guanosine. So it's a deoxy at the number 2 carbon, so it's deoxyguanosine. So the nucleoside monophosphate is deoxyguanosine, because that's a guanine nitrogenous base. The nucleotide, when we still have the phosphate there, then we would name that a little bit differently. We're still deoxy because this is DNA, so it would be deoxyguanolate. So we can also look at all the other nitrogenous bases, and in fact I've got a great picture that I'll provide you all here of all the other nitrogenous bases. So we've been working with guanine and looking at that, but you could have deoxycitylate or deoxyadenylate depending upon which of the nitrogenous bases were involved. So the building blocks of DNA are nucleotides, or you could say they're nucleotide monophosphates in DNA, or they're built from nucleoside triphosphates. And so there's a lot of lingo that goes around that. I want to make note that depending upon the nitrogenous base, the number of hydrogen bonds holding together the 2 strands of DNA are different. So notice that when a guanine pairs with a cytosine, there's 3 hydrogen bonds holding those together, whereas when you see a thymine pairing with an adenine, there's only 2. So that actually is interesting because the A to T bonds are easier to break. A lot of times there's A to T rich regions at the beginning where transcription is just starting off, so that it's easier for the machinery to pull apart the 2 strands of DNA and begin the synthesis of the RNA. So that's what I want to think about now. So as we're returning to the video, and we're seeing, and let's backtrack just a bit on here. So as we return here and we look at the melting of the DNA at the beginning of transcription, melting is one term that you can give to that, or denaturation is another term that you can give to that. So watch here as the DNA is denatured. Okay, so it's pulled apart, and it's pulled apart to enable the RNA to be made. So let's take a moment to look more deeply at that. So what that means when this denaturation takes place is that these 2 strands of DNA will come apart. So essentially the hydrogen bonds here between the nitrogenous bases are broken, and the 5' to 3' and 3' to 5' strands are pulled apart from one another. That is that melting or denaturation process that takes place. So now what happens is with the 2 strands far away from one another within the transcription machinery, this opens up the bottom strand to be used to build the RNA. So if we can recognize the rules of base pairing, that the guanine dictated that a cytosine would be incorporated next, that's the exact same thing that will dictate the RNA sequence as well. So when this guanine is recognized by the machinery making the RNA, it will essentially be the code that says to that machinery, hey, pair me with a cytosine in the RNA. Now, the one thing that's going to be different is rather than having a deoxy derivative added next, we will see the addition of a ribonucleotide, because of course this is RNA. So I'm going to make a copy of the current nucleotide here, recognizing that the RNA that will pair there will look very similar, except for that one big difference is that rather than having a deoxy 2' position in the new RNA strand, that will now be a hydroxyl. So I'm just going to get rid of the DNA strand up here so that we're feeling less confused. Remember, it's denature now. It's kind of off, getting out of the way, minding its own business. It's not part of the process, if you will. And now the RNA is getting made using this bottom strand as its template. So we're getting the incorporation of ribonucleotides, and let's add in the hydroxyl at the 2' position so that we can recognize this as being RNA rather than DNA. Sweet. I'm loving this. You know, just keep noting that this is totally off the cuff stuff. I mean, I'm just building this as best as I can as I go. And that's what I want you all to do this semester, is to try things and not be afraid to fail, because you're going to just be drawing a lot, making a lot of things, and building as you go and being pretty spontaneous. Okay, great. So now what we're seeing is that the 3' to 5' or bottom strand of DNA, the negative sense strand, is making a new strand of RNA. So this RNA is going to end up looking a lot like the plus strand or the 5' to 3' strand of DNA when it's finished, only that you, in every case, will be seeing that it is a ribonucleotide rather than a deoxynucleotide. So that will change the nature of the RNA as compared to the DNA. Additionally, anywhere that there is a T in the DNA, a place where an A existed somewhere on the coding, the template strand, the strand that's encoding for the RNA, that will pair with a U rather than a T in the RNA. So taking a look at those nitrogenous bases once again and recognizing that U's replace T's in the RNA. And really, there's not a big difference, right? We could say that, in fact, thymine is 5-methyl-uracil. 5-methyl-uracil. So it's just that uracil is what we find in RNA. It's the nitrogenous base used instead of thymine, which is seen in DNA. So it's just a methylation difference between the two. Cool. So now we get the synthesis of our RNA. So this is where we found ourselves in the video again, resituating ourselves here and recognizing this process, right? To make RNA, here's the green. The green is the RNA. It's being synthesized from the DNA. So now the challenge that I give you is that RNA, remember, based on the video, the RNA goes on, if it's messenger RNA, and that messenger RNA is converted into protein. So protein is another macromolecule within the cell, and I challenge you now to think about what proteins are made of. Remember, with DNA and RNA, they're made up of nucleotide building blocks, but proteins are made up of a different kind of building blocks. So your extra pre-party question is, what are the building blocks of proteins?